I trip on my own 2 feet, let alone having something in my pacing path here, right? How's everybody doing? How many moms do we have here today? Okay, what do you guys think? Moms get a pop quiz or not? We could compare mom's performance to everybody else's performance. No? You're going to add your score to their grade? No. Okay. So, you guys have big plans for the weekend, moms? To what? Following the lead, okay. And the lead says? Fashion show? I could do a little. You don't want to see that probably, right? Comedy show? Anybody going to the comedy show? Anybody? That's Dad's weekend? Isn't there a comedy show this weekend? I thought there was. Well, to enrich your weekends, I decided to put together a little something for moms today. So, shh. I'm noisy enough. So, these guys have been working very hard on biochemistry, and everything in here has been very serious since day one. So this song, I think, kind of captures all of that. Let's sing loud for Mom. This song, it's called Oh Mommy Dear. Oh Mommy dear, this is my biochemistry. The problem's long, the course is really tough. That last exam, it really put the fear in me. I studied lots, I hope it was enough. But let's forget about it while you're visiting and have some fun when we get out of class. Then when you're gone, I'll go back to my studying. That Kevin Ahern really, truly is an awesome guy. All right. So you can see everything's pretty serious in here. All right. Happy to have you here, moms. I promise I won't make you take a quiz, but I may make them take a quiz. You never know about that. So, the sooner we get out of here, the sooner you can go have fun, right? Yes. So I should talk twice. There's some support back there. The faster I talk, the quicker we'll get out of here. So let's do that. All right. Well, last time I just got started talking about the process of synthesizing proteins. Of course, that's the process we call translation in biochemistry. And I was at the point of telling you what I described as the sort of 0 step or first step, depending on how you want to count. So there's really 3 steps: initiation, elongation, termination. And if we add this first thing I'm going to talk to you about, we can call that a 0 step. Okay? So the first thing that has to happen for translation to occur is we have to put amino acids onto the proper transfer RNA. That's really critical. If we don't get the right amino acid on the right transfer RNA, then nothing else is going to matter. We can have perfectly replicated DNA, we can have perfectly made messenger RNA, no errors of any sort, but if the transfer RNA has the wrong amino acid on it, it doesn't matter, because it's the transfer RNA that is going to bring the amino acid to the point of translation. And if it brings the wrong one, we're hosed. So this 0 step that I talk about, called amino acid activation, is essential for everything that's on the face of the earth, for every living thing that's on the face of the earth. This is one of the, if not the most important step. And I'm calling it the 0th step. Okay? All right. So what is it? Well, during the process of translation, a transfer RNA is going to form a set of base pairs, as you saw the other day, with a codon in the messenger RNA. This will all take place in the ribosome. All right? So that base pairing says, oh, here is the right thing to bring in. That right thing is now going to be an amino acid attached to the other end of that transfer RNA. That means then that we've got to put the right amino acid on. And what you're seeing on this very top of the screen, in very simple terms, is what's happening. Okay? At the 3' end of a transfer RNA, there's a hydroxyl group. And it's onto that hydroxyl group that an amino acid will be attached. Okay? Now, I'm lying a little bit because sometimes they get attached to the 2' end as well. I'll say something about that later. But for right now, at the end of the transfer RNA, that's where the amino acid gets attached. That process requires an enzyme, and that enzyme has a mouthful of a name. It's called an aminoacyl-tRNA-synthetases. I told you I was going to talk fast, right? An aminoacyl-tRNA-synthetases. Say that 5 times fast, and you become automatically a biochemist. That's how I got my degree. Okay? I can talk fast. That's good, right? All right. To keep it simple, we're going to call it Larry. Okay? Larry. You guys know people named Larry, right? All right? The name of that enzyme, for our purposes, is Larry. Okay? They just made it up. Okay? It's simple, right? If you want to call it aminoacyl-tRNA-synthetases, you're welcome to do that. What's that? Let's call it Larry? Okay. All right. So Larry is the enzyme that catalyzes the linkage of the right amino acid to the right transfer RNA. Now, what that Larry has to do is Larry has to read that 3-base sequence that pairs with the codon in the messenger RNA. If you remember what I said last time, that 3-base sequence in the transfer RNA is called an anti-codon. So Larry is reading the anti-codon and saying, oh, here's the amino acid to put onto this transfer RNA, because here's the anti-codon sequence. I know which amino acid that is that goes on here. Last time we saw that the genetic code said that CCC was proline. That means that the anti-codon will be GGG. And so when that Larry enzyme looks at a transfer RNA and says there's a GGG in the anti-codon, it's going to put a proline on the other end of that transfer RNA. Make sense? Yes? It reads the anti-codon because the anti-codon is what's in the tRNA. Okay? So it's got to read that properly and get the proper amino acid on there. It turns out that there are 20 Larry enzymes in a cell. And not coincidentally, there are 20 amino acids that get put onto tRNAs. You might logically assume that there's one amino acid per Larry enzyme, and you would be correct. Okay? So each amino acid has its own Larry enzyme that's going to recognize it and the anti-codon that corresponds to it in the tRNA. Everybody with me? Okay. Well, this simply shows the process of that happening. We're not going to worry about the process of this happening. It starts out unlinked. It ends up linked. Okay? Now, that linkage turns out to be really important and actually kind of interesting, too. Because when Larry puts that amino acid onto the end of that transfer RNA, it's got a problem. And the problem is that all this is occurring in the cytoplasm of the cell. And the cytoplasm of the cell is a very rich environment of water. Remember, we're 75% water. Okay? It's a very rich environment with a lot of water in it. And it turns out that the half-life of that tRNA linked to an amino acid in water is about a half a second. It's not a stable bond in water. Hello, Houston, we have a problem. I need this amino acid to be brought to the ribosome by this tRNA, but the bond that holds it onto the tRNA is not very stable. Okay? Well, fortunately, there is a solution to that. And the solution to that is that there's a protein that covers up that bond between the amino acid and the tRNA so that water doesn't get a chance to see it. Okay? Now, that turns out to be pretty cool. All cells have this. We have this. Bacteria have it because that bond is unstable. Okay? In bacteria, this protein that covers up this bond is very easy name to remember, as you will see later. It's called EF-TU. EF2, you can think of it. EF-TU. You'll see later why that's an easy name to remember. But for now, I just want you to keep that in mind. So as soon as Larry connects those two, there's got to be plenty of EF-TU to be there and cover it up. Because remember, we've got about a half a second in order to protect it from water. Okay? That means we've got to have or in this case the bacterial cell, have to have a lot of EF-TU. And not surprisingly, EF-TU turns out to be the most abundant protein in a bacterial cell. It's that important. Okay? So there's plenty of EF-TU floating around in a bacterial cell just waiting to go and cover up that bond. Putting an amino acid onto a tRNA costs energy. Notice we start with an ATP, we're left with an AMP. Usually we have ATP going to ADP. When we go to AMP, that means we've lost 2 phosphates. It means it's the equivalent of 2 ATPs for every amino acid that's getting put onto a tRNA. So remember I told you that making protein is energetically expensive. This is one of the expenses right here. Okay? There are others. Question? 2 ATPs, that's right. EF-TU is a protein, that's correct, yes. His question was, EF-TU is a protein, not an enzyme, and the answer is yes, it's a protein. EF-TU does not catalyze a reaction. But it does have a function, as I will describe to you. Okay, so this is another different view of a tRNA than what I showed you the other day. The other thing I showed you the other day looked kind of like a clover leaf. And in 2 dimensions, that's actually what it looks like. This is a 3-dimensional depiction of the way that a tRNA actually looks. All right? The place that I was describing to you with the 3 bases is down here. This is the anti-codon. The other end that I was describing to you, where the amino acid was, is way up right here. Okay? Way up right. What did I do? Here we go. I knew I had a pointer here somewhere. There we go. Those are my keys. Okay. So the other end is way up here. All right. So this structure is what's bringing the amino acids to the ribosome for translation. We're not going to worry about all the letters and all the other names and so forth on there. That's the 2 most important regions of a transfer RNA. Okay. Well, prokaryotes have something else that's interesting, and I didn't tell you about this the other day because it was confusing the picture then, but I'll tell you about it now because I don't mind confusing the picture now. I got to confuse the picture at some point, right? Okay. So to confuse the picture for you, I told you that the very first amino acid that makes it onto almost every single protein on the face of the earth is methionine. I lied. Okay? In bacteria, it's a modified form of methionine. It's called formyl-methioinine. Okay? And that formyl group is right there. Okay? No, I'm not going to ask you to draw that. All right? Why isn't it methionine? This is a modified amino acid. Why is it modified? Why does it go on there? Well, it turns out that the synthesis of the addition of the very second amino acid during protein synthesis, the second amino acid, as it goes on, is chemically unstable relevant to the alpha amino group, which is right there. If this alpha amino group is not covered up during the process of protein synthesis, what will happen is that this will form a bond that's not desirable, and translation will stop. So bacteria have evolved a way of keeping that from happening, and they have evolved an enzyme that puts this group on there to cover up that amine group over here. Consequently, this guy can now have an amino acid attached to it to start the process of protein synthesis, and we don't get the undesirable reaction that would occur if this was not covered up. Okay? Now, I'll explain eukaryotes later, but in prokaryotes, that's what's happening. is that we have the very first one is covered up. All the other methionines that go into a protein in prokaryotes do not have this. Okay? Only the very first one. So they've got an N-formyl group that goes on to them. Okay. Now, we're ready to start the process of initiation. And one of the things you're going to see about protein synthesis is that protein synthesis is an incredible mechanism. You guys saw how DNA replication was almost what I described as magical in terms of the things that happened. It was magical largely because of the speed with which it occurred. Protein synthesis is magical because of the complexity of all that's going on. Even though we saw a replication fork in DNA replication that had about, 7 or 8 proteins all working together, the magic of DNA replication was it was occurring at about 1,000 nucleotides a second. Okay? In protein synthesis, we have complexes of proteins, of ribosomal RNAs, of transfer RNAs that are making proteins, as we will see. It goes a lot slower. It goes at the rate of about 15 to 20 amino acids per second, which corresponds to roughly 50 to 60 base pairs per second as it's moving along. So it's a lot slower than DNA replication. All right? But the magic is that this ribosome has about 70 proteins in it. It's got 3 ribosomal RNAs in the case of prokaryotic cells. It's got to deal with 20-plus different tRNAs. And it's got to go sliding along a messenger RNA. So the complexity of this in terms of mechanism is actually more complex than what's happening in DNA replication, so it's not surprising that it goes slower. Okay, well, let's look at the process of initiation. First, I need to tell you a little bit about a ribosome. So I've been mentioning the term ribosome to you, but I haven't told you what it is. A ribosome is a complex of proteins and ribosomal RNAs that translates messenger RNAs into protein. A ribosome is a complex of proteins and ribosomal RNAs that translates messenger RNAs into protein. As we will see, the process of this happening is fairly orchestrated, and that's not too surprising given the number of proteins working there. We have to have everybody playing together nicely, or we're going to have problems. So coordinating this and organizing this is essential for this to function properly. What happens? Well, the process starts as you see here on the screen. First of all, the ribosome has what we call 2 subunits, a small subunit and a large subunit, distinguished only by their size, and of course, which proteins and ribosomal RNAs they have in them. The size, for our purpose, is the most important thing. The small subunit has a name. We call it, in bacteria, we call it the 30S, 30S. And S is an approximation of size. I won't say more about that here. As we will see, it's an unusual approximation of size. The large subunit has a name, and it's called the 50S, 50S. Okay? When the 2 get together, it makes a complete ribosome that you might think we would call the 80S, but in fact, its size is 70S. You see why the approximation of size is odd. The sizes are not additive. That is, the S unit is not an additive unit. But 70 is bigger than 30 and 50. Okay? All right. We start with a 30. A 30S ribosome subunit, the small subunit, has a bunch of proteins, but most importantly for our purposes, it contains a ribosomal RNA that performs a critical function. It's called the 16S ribosomal RNA. You guys are getting tired of all this S, this BS, I guess, right? BS. Bad joke. It's Friday. Okay. A 16S is the ribosomal RNA that we find in the 30S small ribosomal subunit. Okay? Now that 60S subunit, as I will show you in a minute, has a very cool sequence in it called, it has a name, I'll give you that later. I want to throw it up right now. Okay? The name's right there if you're curious. All right. What happens is, the first step of the process, we have a linkage between the messenger RNA, which is the blue guy over here, and the small ribosomal subunit, which is here. Now, you've been thinking about translation. You say, well, the ribosome's got to move along. It's got to translate that sequence from one end to the other. But it turns out that it doesn't start at the end and then just move in. Okay? It starts at an AUG. Remember, the AUG is the first codon that corresponds to the first messenger RNA. You say, oh, well, that's simple. It just goes to the first one. Well, it turns out that there are other AUGs in a messenger RNA, and it's not clear where the translation should start. The cell's got to have a way of knowing where does translation start. And that's where the 16S ribosomal RNA comes in. Okay? The 16S ribosomal RNA has a sequence that's complementary to, that is, it makes base pairs with, a region of the messenger RNA called the Shine-Dalgarno sequence. Shine-Dalgarno sequence. So the Shine-Dalgarno sequence is found in the messenger RNA, and it's complementary to a sequence that's in the 16S ribosomal RNA that's in the small subunit. They form base pairs. Why is that important? That's important because this 16S ribosomal RNA is in a specific place in the ribosome. And what it's doing is it's aligning and telling the ribosome this next AUG that you see is the place to start. This is the place to start. So the Shine-Dalgarno sequence makes sure that that ribosomal RNA is lining up the messenger RNA properly so that translation starts in the right place. If we start translation in the wrong place, we're going to make the wrong protein. All right. Well, there are some proteins called initiation factors that help this to happen. Initiation factors are abbreviated IF. See, moms, I told you this was complicated, right? They're called IF. And they have numbers. Initiation factor 1, initiation factor 2, initiation factor 3. And no, we're not going to break them down, which one does which, except for I'll tell you what initiation factor 2 does. Initiation factor 2 brings to this complex that's building, it brings that very first amino acid, which we said was F-met. Right? F-met's going to be the very first amino acid that's going to appear in this protein, and it's brought there by IF2. IF2 does for initiation what EFTU does later. IFTU is protecting that amino acid, that very first amino acid on that transfer RNA. This is the only one that's bound by anything except for EFTU. All the other tRNAs are covered up by EFTU. Should I slow down? Oh. Breathe. Remember to breathe. Initiation factor 2 brings the first amino acid to this. What we're doing is we're building a complex. Initiation factor 2 is bringing that first amino acid to that complex.
Which is just the small subunit
right now. Right now it's the small subunit, it's the messenger RNA, and we're going to see the large subunit that's going to come into play in just a minute. Questions? Yes. His question is, does IF2 also protect the F-met from water? It does. So IF2 is performing the identical function that EFTU is performing. What we'll see during the process of elongation, EF stands for elongation factor. And that 2 corresponds to 2, right? So the EF2 protects all the amino acids except for the first one, and IF2 protects the first one. Can I go forward? Questions? I'm happy to answer questions. If you don't answer questions, I'm not going to slow down. If you don't ask questions, I'm not going to slow down. There's a question. What does EFTU cover up? Okay, so remember that when we had Larry catalyze the enzyme, the reaction between the amino acid and the tRNA, okay? The bond between the amino acid and the tRNA is unstable. That's what EFTU covers up, okay? The one exception to that is IF2 is covering up the bond here on the FMet. Okay. I'm going to dive back in. Okay. So, what have we had happen so far? We've had the messenger RNA has bound to the small subunit. An initiation factor has brought in the first amino acid on the first transfer RNA, and the messenger RNA has been linked to the proper place on the ribosomal subunit. We've aligned things. Okay? You'll notice that what IF2 has done is it has brought this guy in and it has lined up that transfer RNA so that base pairing has happened. Notice there's the AUG, there's the UAC that forms the base pairs with it. Everything is lined up to get started because all the action is going to occur right where this guy is right now. It's all lined up. It's all ready to get started. We just have a couple of other things to bring in. Well, excuse me. Well, one of the other things we have to bring in is the large subunit. Here it gets easier, actually. The large subunit is brought in by other initiation factors, IFs. Okay? You can call them IF1, IF3 if you want to, but suffice it to say that other initiation factors help bring in the large subunit and put everybody together. Okay? At this point, the ribosome is there together. So we can think about this, if we look at this, as I like to think of the ribosome as kind of like a snowman. A snowman has a big bottom, and a smaller top, right? So that's what a snowman looks like. In between the two balls of the snowman, that's where the messenger RNA is. It's sitting in between the two. And it's inside of this snowman where the protein is going to be made. Inside, between my two hands, that's where the amino acids are going to be joined one to the next. Bear with me? Okay. We have reached the point where we have finished initiation. Initiation is now done. Okay? Now it's time to elongate. Right? Oh, before I do that, let me show you the Shine-Dogarno. Okay. I said there was a sequence called the Shine-Dogarno sequence. If we look on a messenger RNA, that's where we see the Shine-Dogarno sequence. We commonly see a very familiar sequence. Here it's GGAG, GGAG, GGG, UGG, AGGA. They all are within about 10 bases of the AUG, the start codon. It's this sequence, the Shine-Dogarno, that's complementary to a portion of the 16S ribosomal RNA. Okay. So the Shine-Dogarno is there. If we look at it, it looks something like this. And on average, if we average it out, the average sequence is this guy right here, GGAGG. And no, you don't need to memorize that. But you should know what a Shine-Dogarno sequence is and where it's found. Okay? A Shine-Dogarno sequence is found in a messenger RNA within about 10 nucleotides of the starting AUG. Okay, let's elongate. Lovely big figure there. I hope it's not too big for you. All right. During the process of elongation, what's going to happen? The ribosome is going to read the next codon. It's going to bring in the right transfer RNA with the right amino acid on it, and then the amino acids are going to get joined to each other because we're synthesizing a protein. That's the big picture of what's going to happen in elongation. Okay? I'll repeat that. All right? So what's happening is the ribosome is going to read the next codon. Remember, it's already read the first one, which is AUG. It's going to read the next codon. It's going to get the right tRNA with the right amino acid, and it's going to join the amino acids. So we're starting to synthesize a protein. All right. Now, this isn't too bad. When we look at the ribosome, we discover that the place where that very first tRNA was has a site that has a name called the P site. The P site. So that very first F-met tRNA is sitting in the ribosome's P site. There are two other sites. There's an A site and there's an E site. E stands for exit. That's where everything's going to go out. Everything's going to come in on the A site. And everything in the A site will eventually become joined to whatever's in the P site. Okay? All right. Well, here's where we finished. The very top slide shows where we finished initiation. We've now got to elongate, and elongate means we've got to bring in the next transfer RNA that's complementary with an anti-codon to the codon sitting in the A site. So the A site has a codon in the messenger RNA that's sitting there. We want to find the right tRNA that forms a base pair with it because it's going to have the right amino acid on the end. Well, guess what? You saw in the initiation phase that IFTU brought in that AUG, the F-met-methionine. EFTU, which you've already seen, is going to bring in this one. It's going to bring in all of the other tRNAs. So EFTU, which is covering up that bond, in addition, helps get the right tRNA into the A site. Okay? Now we see that the right one has been brought in. So we've got the first one sitting in the P site on the left. We've got the new one that's in the A site on the right. Each one has an amino acid. There's an F-met, and there's the next amino acid. Then we join the two with a peptide bond, a covalent bond between that first amino acid and the second amino acid. That leaves the two of them attached to the second tRNA. The first tRNA at this point is empty. The next step in the process requires energy because the ribosome has got to slide down one more codon to put a new codon in the A site. It slides down 3 nucleotides down the messenger RNA. That moves the previous tRNA into the E site. It's getting ready to exit and leaves an opportunity for a new tRNA to come into the A site. Yes? Oh, very good question. His question is, when does the EFTU leave the tRNA? Very good question, okay? We don't want it to leave too soon because what's going to happen? Water's going to break it, right? So once it gets in the confines of this ribosome, there's no water, and EFTU leaves. The inside of the ribosome is free of water. That's cool. It's important because we don't want that bond to be broken there, right? Don't want that bond to be broken, okay? I'm going to tell you 2 more things, but then before I do that, I'm going to ask you for questions because I don't want to go too fast. This is deep stuff. I roll my pants up here. Yes? Very good question. So this on the left is the 5' end. The 3' end is over here. So translation proceeds from 5' to 3', just like you've seen everything else. So during the process, this is going to be sliding to the right. Okay? Yes?
When it moves over, because I can't see.
Yeah, it's a small figure. That's why I like you to print them out beforehand.
When it moves over, then the second one actually starts taking the P spot?
Yes.
And
Yes.
the first one actually takes the
Yes. So her question is, when this slides over, does everything slide over as well? And that's correct. So what was in the A site slides over to the P site. So everything's going to slide over one, or 3 bases as it were, and things that were there are going to slide with them so that what was in the A site becomes in the P site, what was in the P site moves over to the E site, and now we've got an empty place in the A site for a new one to come in. Clear as mud? Man, on Friday, Friday's the worst day to give this lecture. Everybody is ready to do something besides this, right? All right, I said, yes.
So the EFTU helps bring the right amino acid to the A site?
The EFTU helps bring the right amino acid on the right tRNA to the A site. That's correct. So EFTU helps that process to occur. In addition to covering things up, it's helping to get the right one in there. Okay? Two things. All right? Two things. One thing, I didn't tell you what made the peptide bond. You say, oh, it's an enzyme that made that, right? No. This is the first example that you will see of something called a ribozyme. A ribozyme. Enzymes are proteins that catalyze reactions. Ribozymes are RNAs that catalyze reactions. Ribozymes weren't even known until about 25 years ago. And when they first discovered them, it was like, holy crap, never dreamed that an RNA could catalyze a reaction. Well, not only does it catalyze reactions, it catalyzes the most important reaction that's going on inside of you, the synthesis of proteins. Remember that the ribosomal subunits have ribosomal RNAs inside of them? That's what's involved in that catalysis. It turns out there's a ribosomal RNA in the large subunit called the 23S. And that is a ribosome, a ribozyme. Ribozyme, see, I'm doing it too. It's easy to say ribozyme, ribosome, enzyme, right? Okay, they run together. A 23S ribosomal RNA is a ribozyme. It catalyzes a reaction, and the reaction it catalyzes is the peptide bond. The other thing I'll give you is just a name. This one isn't as complicated. And that is, I didn't tell you what caused the ribosome to slide down 3 more bases. That's actually another elongation factor, and it's called, this is very easy, EF-G. EF-G catalyzes the sliding down of the ribosome. It's what we call translocation. In the song we sang the other day, I talked about translocation. That's what's happening here. Interestingly, if we look at translation, we see that the energy source is not ATP. The energy source here is GTP. GTP is the energy source in translation. All right, we're almost there. We've got to terminate yet, but termination turns out to be pretty simple. Questions before I move forward to termination? Okay, let's make it into termination. Okay? Now, this ribosome has been moving along. It's got a whole bunch of amino acids that have been all joined together. There's that protein that's being made. It turns out there's a little tunnel that comes out of the ribosome. It's kind of cool. It comes out of a little tunnel in the top of the ribosome. Okay? What happens when the ribosome moves along and what appears in the A site is a stop codon? We talked about stop codons last time. Well, it turns out stop codons aren't complementary to any of the tRNAs. There's no tRNA that's going to form base pairs with it. All right? So it turns out that there are, I can't say the word, there are factors that help to terminate this process. Okay? They're called RFs. Okay? RFs. RFs recognize that a stop codon is in the A site. And they actually will insert themselves into the A site, and they bring one thing to the A site. And that one thing causes the protein to be released. Anybody have any idea what that one thing is that they carry? Water. They carry one molecule of water. Did you say that too? Good. You get a gold. Did she say it or did you say it? You get the gold star for the day, okay? They carry water. Because that's all it takes is a molecule of water to add to this thing. It breaks the bond that was holding everything together, and now, as you've seen before in transcription, everything falls apart. That is pretty cool. So I tried to convince you that water was very critical in this process. At least one person who's not even a student in the class picked this up, okay? So water is critical, right? And as a consequence of that, right, the protein gets released. We've just finished the process of translation. Yay. Hey. Take a bow, yeah. All right, give the ribosome to take a bow, right? Everything goes away then. That's kind of cool. All right? Now, this is an intricate process, all right? This is occurring, as I said, at the rate of about 15 to 20 amino acids per second. All the steps I just described to you take about 15 to 20 amino acids per second, which is about 50 to 60 nucleotides per second. It's moving along with it. That sounds slow compared to DNA replication, but for all the things that are happening, that's pretty good. Pretty good. Questions? Yes? Okay. Oh, very good question. How far behind this ribosome is the next ribosome? It turns out that when translation is occurring, there will be multiple ribosomes, one following the next on a complex like this. Probably within about 50 nucleotides or so, you'll see the other ribosome sliding along. Yeah. If you think about it, having multiple ribosomes on a messenger RNA is good because it allows more protein to be made per messenger RNA and faster. Yes? Good question. Are there errors that occur in this process? And I think the next question is there a proofreading that occurs in this process? All right? A very good question. Okay? Proofreading was pretty important for DNA replication, right? Because we didn't want mutations to happen. Because mutations will be passed from one generation to the next generation. If a mistake happens during, let's start with transcription, it doesn't have the same consequence as if a mistake happens during DNA replication, because remember, messenger RNAs get degraded. They're not around forever. When we look at the process, what we discover is that transcription has a greater error rate than DNA replication does, reflecting the fact that it's not quite as critical. It's not high, but it's not as good as DNA replication. When we look at translation, which is her question, we see an even higher error rate. But again, proteins aren't around forever. If the cell makes a mistake in a protein, maybe the ribosome following behind it won't make that mistake, okay? And everything will be fine. So the consequences of making an error actually decrease as we move farther in the central dogma. Protein mistakes are of less consequence than RNA mistakes that are of less consequence than DNA mistakes. Okay? Yeah. Yes, Olivia.
When the ribosomes make it to the poly-A sequence,
Yep.
do they code that as well?
Okay, so our question is, if a ribosome makes it to a poly-A sequence, does it code that as well? The answer is no, it does not, because before then it will have hit a stop codon. So if we think about it, it's a good question. If we think about a messenger RNA, I told you we didn't start at the 5' end because we start at the first AUG, which is not the end of the sequence, it's actually internal. And the stop codon is before you get to the poly-A sequence. So the far 3' end and the far 5' end aren't read by ribosomes. Okay? All right, let's do just a couple things relevant to this, then we'll do a song. How about that? Okay. It turns out that protein synthesis has some very, it's pretty critical. Every cell alive has to make protein. If you can't make protein, the cell's dead. And that gives us a way to kill cells. So one of the biggest targets for antibiotics is the process of protein synthesis. Because if you can stop protein synthesis in an organism, you can kill the organism. And if we can stop protein synthesis in a bacterium without harming our cells, then we've got a really nice antibiotic. Well, there are many things that will stop protein synthesis specifically in bacteria without affecting our cells. Okay? So one of the really good examples, actually it's not up here, but one of the really good examples is everybody's probably had tetracycline. Tetracycline stops protein synthesis only in bacteria. Okay? Some of these things, like puromycin, may affect both bacterial cells and your cells, so you probably wouldn't want to take a little bit of that, you know. Not a good idea. We use them as laboratory tools to do things, right? But it wouldn't be good as a general antibiotic. But many of the antibiotics, in fact, are specific for bacteria. Chloramphenicol is another one. Chloramphenicol is specific for the process of translation in bacteria. Yes? Yes. Yes. Okay, so penicillin allergy. The allergy component relates to your immune system recognizing something as foreign. Okay? So that's what a penicillin allergy is. Penicillin is one of the antibiotics that doesn't affect translation. So it actually affects the synthesis of membranes. Okay? So not all antibiotics affect translation, but many of them do. But an allergy to any antibiotic relates to your immune system recognizing it as foreign. Okay? All right. The reason I show you this figure is if we look at puromycin, puromycin looks a little bit like an aminoacyl-tRNA. That just simply means the tRNA attached to an amino acid. So this guy fits into the A side of the ribosome nicely. And when it gets in the A side of the ribosome can't do anything with it. And it's hosed. So basically what puromycin is doing is it's stopping the process of elongation. If you stop elongation, you stop protein synthesis. Another target, and you could probably imagine some of these yourself, we say, oh, okay, well, bacteria have this F-met, that putting that formyl group happens on bacteria, but it doesn't happen in eukaryotic cells, as we'll see. If we can target that F-met, we can stop the process of initiation, and in fact, some of the antibiotics actually do that. They stop the addition or the synthesis of F-met. They don't affect us because we don't make F-met. We have different mechanisms that we use, and we can kill bacteria that way. So part of this is, there's a lot of complexity in what I'm showing you. But it's important to recognize that what we learn by looking at complexity is we can figure out ways to do cool things. And that's why an understanding of these is important. Yes?
Is there any reason you would use something that stops initiation or something that over something that stops elongation?
Question is, would there be a reason to do initiation versus elongation versus something else? Basically, no. What you're going to care about if you've got a throbbing infection that's killing you, all you care about is stopping that process in general. So something that maybe you've got a penicillin allergy like she's talking about, doctor may say, okay, well, tetracycline will work well. Let's put tetracycline in there instead. Okay? What do you guys say to a song to finish, right? Okay, let's have a song. So this one, you're welcome to sing. I can't sing it because I can't sing it worth a crap. And I've got a very good professional recording of this song. So how many know the song Maria from West Side Story? All right, well, here it comes.
Translation.
Ah.
The most intricate thing I ever saw. From 5 prime to 3 prime. The final step that we know about the central double.
You can sing along, please. All right, guys.